Only have to take photographs and you have to submit it. Yeah. So I was expecting this question from at least one of y'all, and I got the question that means how was the NEET 2021 paper? So as far as yeah, homework will not be assigned. It's already told to you all, so you only have to submit it. Okay, if I assign now, then you have to give it in the right time. So it has not been assigned as such. Okay, you just submit it. You have to submit the photograph. Huh. Yeah. Share to all. Yes, you can. Okay. PDF is, yeah, PDF. You have to make a PDF of the photographs that you have taken and you have to submit it. Okay. You will be told if you are not getting it now. I'll tell you later on. Okay. That how to do it. Right now, focus on the lecture. Okay. Yeah. So firstly, the bio paper, as expected, it was really very nice. And as I have continuously been telling you all right from day one, that please prepare your NCRT very thoroughly. As expected, out of the total questions that were asked for bio, that is 50 for botany and 50 for zoology, there were 100 questions that were asked. Out of these, only two questions were not direct questions from NCRT, but these have been already taught to you all. Okay, so the extra content that I always take, like during the lectures only, I give you all so many extra points. My presentations you have seen already has got so many extra points. So those all we have considering all those things now. Even those two were very simple questions. So it wasn't difficult, but it was a lengthy paper. There were almost more than 15 match the columns. Now what happens in match the columns, beta? In match the columns, they are asking you 4 options. I mean, they are writing in column A 4, in column B 4, right? So they test the knowledge of 4 things for 4 marks. If it's a one-liner question, then what will happen? For that one line, you will be given 4 marks. In match the following, what happens? You should actually, if they make one sentence question on those 4 particular things, then you could have got 16 marks. But that is the reason they have combined 4 points together and they have made match the following. Of course, the match the following was also very easy. Everything was based. So I would say 98% of the paper, out of 100 questions, 98 questions were NCRT, line to line NCRT. So definitely it was a very, good paper. There was no problem with the paper at all. So, but match the following is going to take time, and they had almost 8 to 10 questions wherein you have to select correct, incorrect statements. So when they ask you correct, incorrect statements, now again you have to read 4 sentences. So reading में time ज़्यादा जाता है बच्चों का. One liners फटाफट solve हो जाते हैं, but to read the match the followings, to read the correct, incorrect statements, it's going to take time. So that particular practice needs to be given. Okay, otherwise the paper was really good. As far as bio is concerned, I have like certain students who are getting 360 on 360 complete, 355 many students are getting, 350 plus also many students are getting in bio. I'm very sure about it. As expected, again, physics was a bit tricky. It wasn't difficult, it was a bit tricky. And chemistry was also almost same like last year. So it wasn't like like when you study, it's not going to be difficult. Okay, what should we attempt first? Biochemistry or physics? The subject which you are confident with, you attempt that. If you are confident with physics, go for attempting the physics question paper first. If you are confident of your preparations for chemistry, go for chemistry. If you are perfectly okay with bio, you're comfortable with bio, go for bio. It's your individual choice. I won't say you go for bio, you go for chemistry. It's your individual choice that which particular subject have you mastered. So depending on that, you select your particular subject and go ahead with that. Was of the old paper pattern or new section A and B? A and B. A and B. They have already told A and B sections. Okay. Are all questions compulsory to attend, or we can leave the ones that we don't know? You can leave the ones which you don't know, because otherwise you will get negative marking. But out of the 50 questions, question number 1 to question number 35 are compulsory. And of course, compulsory means you don't have any option over there. If you don't know, you can leave it. But question number 36 to question number 50, you had 15 questions, out of which you could solve any 10. So that same paper pattern will be there for you all also, hopefully. But I have already started giving you all the question paper for 50 questions right from the beginning, right? So for these students, it was a little bit of, task to read 200 questions in 180 minutes. Okay, it was for the first time. Otherwise, it used to be 180 minutes and 180 questions. But for the first time, it was like 180 minutes and 200 questions. So they did not get enough practice. But as far as bio is concerned, right from day one, when you had joined masterclass, from that day onwards, I have given you 50 MCQs ka practice, which is going to go on. And I'm not going to give you options. The optional wala part will be the last exam just 15 days before your examination. So no options in case of my papers. Okay. As of now, so not at all. Later on, just few days before your actual examination, you will get a test series wherein you can leave the questions. Not now. Yeah. That is because of Ganesh festival. Na, many people were not there or something is what I came to know. So that was the reason. Because of carbon paper. Why? We'll have reproductive health or reproduction living organisms as well. The topic she will Preeti ma'am will send you. She will send you the topic. It's going to be health, if I'm not wrong. Reproductive health. Okay. It will not come as to do. It will not come as to do. I'll tell you all. Okay, I'll tell you all how to go about it. Answer has not been printed. That 90 question answer. Which 90 question answer has been not printed backside? I'm not getting it. Which 90 question? I'm not getting your question, beta. Which is that 90 question? Yeah, you have to share the homework with the whole class. Share it. In neat paper, the carbon paper was default. That is there. That is there. That is there, I know. That happened. But that's okay. That doesn't matter. The actual OMR sheet is going to be assessed. Na? Okay. Well, my dear, so what is the highest predicted till now? As far as bio is concerned, 360 and 360 is highest. One of my, student is getting full. There are many with 355. You'll get that I'll tell you what is carbon sheet and everything. I'll tell you when you are going to actually attempt your neat paper. All the instructions and all, like a alag se seminar will be there for guiding you all for how to go about it. So don't worry. Okay, fine. So let us come back to our chapter. That's going to be, we have started with sexual reproduction in flowering plants, and in the last class we had completed till microsporogenesis and male gametogenesis, right? That they will inform you, beta. MAT hai, ya DAT hai, ya VAT hai na, Preeti ma'am will inform you. Can you please clarify which structure is haploid and which is diploid among the microspore nucleus, vegetative cell and germinative cell? Okay, see, first thing, microspore nucleus means microspore is going to be pollen grain. So pollen grain is a product of meiosis. So after meiosis, the nucleus is always going to be haploid, right? So microspore ka nucleus is going to be haploid. Vegetative cell is going to be the mitotic product of the microspore. So it has got only one set of chromosomes. So after mitosis, it's going to have only one set. So even the vegetative cell is going to be what? Even the vegetative cell is going to be haploid. And what is the next one that you're asking? Wait, don't put any comments, Beta, because otherwise that question is going up. Hmm. Huh. Generative cell. Yeah, the generative cell is also a product of what? Mitosis of the haploid nucleus. So even it's going to be haploid. So microspore nucleus, not the microspore mother cell. Microspore mother cell is yet to undergo meiosis. So that will be 2N. That's going to be diploid. Microspore nucleus will be haploid. Vegetative cell is going to be haploid, as well as generative cell is going to be haploid. Okay. You told to, yeah, I had actually, yeah, actually I had planned this, but then there was, I even had a word with the administration department regarding keeping this introductory lecture for you all that time only, but then there was some extra lectures or something that I came to know that you all were busy that Saturday, so we couldn't keep it. So let's see when you all are going to be free, we'll try to organize that. I mean, our both timing should match. Even you should be free and even I should be available at that time, then we can plan out that. Okay, new paper pattern again. A new paper pattern is very simple. So you have got question number 1 to 35 compulsory. You don't have any options for that. And 36 to 50 is going to be optional. Out of 15 questions, you can attempt any 10 questions. That goes for all the 4 subjects. 4 subject means physics, chemistry, botany, zoology. Okay. Then what else? Paper pattern. They divide mitotically. Yes, correct. Generative cell is also going to be haploid. Right, Beta. Paper pattern on group, you will get. Don't worry. Not right now, don't worry. So in the optional questions, there will be no negative marking. Optional question may you can leave any 5, your marks are not going to be deducted. But if the 10 that you have selected, the 10 questions that you have selected, and if you go wrong with that, you will have a negative mark. Okay, that's going to be there. Don't worry, I'll explain the entire system of the paper pattern, of the frequently asked questions, everything in detail once you all are going to be in that particular, after 1.5 years. I won't say 2 years because your exam will be in the month of May only. It's not going to get prolonged till September. So it's going to be 23 na? 23 May. Which part secretes the wall held record? Yeah, today I'm going to talk about it. Will we get negative marking if we attempt the extra question? No, beta, you won't get a negative marking for the extra questions, but what it is going to be, like suppose there are 15 questions, which are going to be optional. Out of 15, you can attempt any 10. So if you attempt 1 to 10 and if you attempt 11, 12 also, and suppose your second answer goes wrong and your fifth answer goes wrong. So what you would feel, I have done till 11th and 12th, so I should get my, all marks corrected. No, it's not going to be like that way. The first 10 that you are going to attempt now will be considered. वो wrong है, right है, कोई फर्क नहीं पड़ेगा. Got it? So that is going to be a very important part. That also I'll explain. Don't worry. Okay. Like it would be in September. No. September is for this year. September is going to be for this year. Not for the next year students also will be, like the ones who are in 12th right now, even they would be giving the exam in the month of May. So you all are definitely going to give in the month of May. First Sunday of May would be your exam. Okay. So don't take that in your mind that you'll be getting extra time to, till September, October, even you will get time to study. No, you have to be prepared right from today. Okay. CET is only for pharmacy. Total questions in all subjects are 50, 50, 50, 50. 50 physics, 50 chemistry, 50 botany, 50 zoology. Out of which you have to attempt 45, 45, 45, 45. Out of 45, first 35 are compulsory. Okay. I'll explain that when the time comes. CET is going to be in the next week, not now. But you will be having it in the month of May only. Till your time, God knows till what patterns are going to change and what things are going to change, you really don't know. So right now, there is no point discussing all these things. What's your doubt? Vegetative nucleus is first, and then second, there are two male gametes in the pollen tube. Microspore nucleus will divide to form one vegetative cell and one generative cell. The generative cell will undergo one more mitosis to form two male gametes. Right? Okay. For example, I feel you have to have that much of confidence and practice, beta. Should have that much of confidence and practice. You have got total 38 chapters to do. 4 से क्या होगा? Huh? Okay. So let us move ahead with that's going to be, we have completed with the microspore and we're going ahead with the megaspore now. So the pistil, the megasporangium, that's going to be the ovule, and the embryo sac is what we have to understand from today's lecture point of view. Okay. The process of formation of megaspore from megaspore mother cell by meiotic division is known as megasporogenesis. Megaspore, नाम से पता चल रहा है बेटा, mega means large, right? So it's going to be larger in size. It's going to be the female gametophyte. I have already told you what do you mean by the word gametophyte. Gametophyte means to hold, to produce, to make, and to sustain that particular structure. That is your phyte. So here is going to be megasporophyte. So this particular one is going to produce megaspore. This process takes place in the ovule. So we should first understand the entire structure of the ovule for us to understand what is this megaspore all about. Okay. So structure of the ovule or the megasporangia. Ovule is considered to be an integumented megasporangium. Please pay attention to the language, my dears. Otherwise, ये सारे bouncer हो जाएंगे. समझ लेना इसको, बहुत सिंपल है ये, बहुत ही ज्यादा सिंपल है. यहाँ पर what you have to see first, I'll go from the basic, that is when you look at a flower, okay, the flower is going to have calyx, which is made up of sepals, which we have done earlier, corolla, which is going to be made up of petals, right, androecium, which is made up of stamens, right. Stamen is made up of anther and filament, right. Anther will be having anther lobes. Each anther lobe will generally have two microsporangia or the pollen sac. Inside the pollen sac, there is going to be the pollen grain, is what is the general idea that we have seen. In the same way, after we come to the megaspore, we have to just look at the fourth whorl of the flower, that's gynoecium. This gynoecium is going to be made up of what? A carpel, or it's going to be called as a pistil. What is this? जैसे stamen था ना बेटा, वैसे ये pistil है. So as stamen is made up of anther and filament, वैसे pistil is made up of stigma, style, and ovary. These things we have already seen. I'm just repeating and giving a recap of all the things, right? The ovary is going to contain the ovules. The ovules in the earlier stage are going to be called as megasporangia. So now you focused. How are we going ahead? Did you understand? So flower ka fourth whorl is gynoecium. Gynoecium is made up of pistil या फिर carpel. Each pistil is made up of stigma, style, ovary. Ovary के अंदर है ovules. इधर तक समझ में आ गई बात बेटा? तो जैसे फ्रूट के अंदर आपको सीड्स दिखाई देते हैं ना, वैसे ओवरी के अंदर ओव्यूल्स होते हैं. So after fertilization, once the fertilization is taking place, the male gamete and the female gametes will fuse together. After that, the ovary will get converted into a fruit and the ovule will get converted into a seed. तो जो भी आप fruits खाते हो ना, it is you are eating the ripened ovary. और जो seeds आप निकालते हो ना, that is the fertilized ovule. Okay? Okay? Megasporangia is same as ovule. Earlier stages, yes. Initially we call it as megasporangia. Okay. So now you understood. Now can you imagine these things? Huh? The fruit that you eat, mango. The mango that you're going to eat is what? It's actually the ripened ovary. Fine. And the seed that you're going to remove is what? It's the fertilized ovule. Yes. Ovary forms the fruit and ovule forms the seed after fertilization. Okay? So we are looking at that particular structure which is eventually going to become a seed. So उसके पहला pre-life जो है, that is the ovule we are talking about. Fine. So they are saying it is integumented megasporangium. Integument. What do you mean by the word integument? Integument means envelopes, covering. So the ovule is going to have coverings over it. It's going to have envelope over it. Okay, got it? After fertilization, these integuments will get converted into seed coat. Okay, आपको पता नहीं, मुझे आपने ये कभी देखा है कि नहीं, बट अपना नींबू की सीड या फिर ऑरेंज की सीड जो होती है ना, वो थोड़ी ड्राई होने के बाद है ना, इफ यू ट्राई टू पील इट ना, यू कैन गेट दैट कवरिंग आउटसाइड. एक कवरिंग निकल आता है थोड़ा ड्राई होने के बाद. मुझे पता नहीं आपने ये कभी टाइम पास किया है कि नहीं. बचपन में मैंने ये टाइम पास बहुत किया है. वो सीड, yes, 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 correct, correct, yes, Bhumi, right, beta. So you can actually see the chilka coming out seed के ऊपर का. Okay, why that? Do you eat sprouts? मूंग खाते हो बेटा? मूंग खाते हो? Sprouts. How do you eat it? Directly खाते हो? मूंग को लिया, मुंह में डाला. You soak it, exactly. Very good. Yes, yes, yes. You soak it overnight, right? And next day morning है ना, उसमें से पानी निकाल के उसको आप बंद करके रखते हो कपड़े के अंदर, right? And then in the night when you see, you can see that white color का thing that is popping up, and you can actually see the green color का छिलका coming out, है ना? Correct? So वो जो छिलका निकल आ रहा है ना बेटा, that green color का coat जो निकल आ रहा है ना, that is the seed coat. But when that seed was an ovule, it was not fertilized. It was called as integument. So you understood what is an integumented megasporangium का मतलब समझा बेटा? Covered ovule, enclosed ovule. ठीक है? First sentence clear? I'll tell you radical and femule. I'll tell you. Don't worry. The ovule consists of stalk and the body. So the ovule is going to have a connector, a stalk, and the main body, right? Two parts of the ovule. The stalk is going to be called as the funicle. You don't have any idea what I'm talking about. You haven't tried. You don't eat sprouts. Krishna, you don't eat sprouts. Oh my God. Oh my God, that's really surprising. I guess you are a vegetarian or a non-vegetarian. Everyone is going to eat sprouts at least sometimes in the life. Yes or no? If not, you do one thing. Yeah, yes, Bhumi. Arey, you do one thing. You go to the market, get 100 grams of moong from the market. Soak it today, overnight. Huh? Tomorrow morning, let the water, drain out the water and keep it in an enclosed, डब्बा या फिर उसके ऊपर ढक के रखो. And then you can actually see the green color का छिलका coming out. I'm surprised. I'm really surprised that if you have not eaten these. Oh my God, sprouts तो सब लोग खाते हैं. Anyways, So try it out. मूंग है, मटकी बोलते हैं हम लोग उसको, ब्राउन कलर की, चौली होती है, बीन्स होते हैं, कौन से भी स्प्राउट्स ले लो। छोले खाते हो, छोले, कि वो भी नहीं खाते हो? एटलीस्ट यू ईट छोले, राजमा, हाँ, इफ यू एटलीस्ट ईट दैट, हाँ, इफ यू एटलीस्ट ईट दैट, देन आफ्टर कुकिंग ऑल्सो ना समटाइम्स वो छोले का छिलका आ जाता है मुंह में। हाँ, एग्जैक्टली, इट्स वेरी हेल्दी फूड। हाँ, so that coating, that covering is what I'm talking about. I think till in my entire 20 years of teaching career, you're the first one who said that you don't know what are sprouts. I mean, I'm really surprised. Anyways, so okay, good. You should eat all these sprouts. They're very good for health. They're rich in proteins. So the stalk of the ovule is going to be called as the funicle. Okay. One end of the funicle is attached to the placenta and the other end to the body of the ovule. Now, what is placenta? Placenta is going to be a nutritive tissue. Okay. It's going to be a nutritive tissue. Nutritive means it's going to provide what? Nutrition. So the developing ovule requires nutrition. So for nutrition, it's going to be attached to the placenta. So just now in the last month, we had studied about the human reproduction. So we know what is placenta and human reproduction chapter. We have already learned about it, right? In the same way, here also, the placenta is going to be for what? The placenta is going to be for, yeah, for the nourishment of the ovule. Yes, but exactly like, you're right, my dear Vaishnavi. So just like how the umbilical cord is going to be present in humans, in the same way, this funicle is going to be present in plants, right? Very good. Perfect answer. Well, the point of attachment of the funicle with the body is called as hilum. Okay, what it is going to be called as hilum. So just like the navel part where the umbilical cord is going to be attached, in the same way, here is going to be the hilum. So hilum is the point of attachment of the funicle. Understood what is hilum? Any difficulty in understanding these points? Funicle is the stalk of the ovule. Hilum is the point of the funicle with which it is attached with the body of the ovule. Okay. Sometimes funicle gets fused with the body and of the ovule, and it's going to be towards one side. It will lie towards one side, and it forms a ridge, which is going to be called as raphe. Remember this word raphe. Huh? Raphe क्या होता है पता है? So sometimes, like you might have heard about this, that sometimes when the deliveries of the babies are going to be via a C-section, cesarean. Why? Because the umbilical cord went around the neck of the baby, right? You have heard about this, right? But it doesn't get fused with the neck of the baby. It is only going to go round. In this case, now this funicle will get attached, means it will become one unit. It will act as a single unit. It won't be separate. So if that particular, attachment takes place to form one single unit, then that unit will be called as what? Raphe. I'll show the diagram. You will understand that better then. Yeah. So here you can see in this particular diagram, you can very well see. So this is the pistil or the carpel as we call it. This is going to be the stigma, right? This is the stigma. This is going to be the style. This is going to be the ovary. And at the base, you have got thalamus. Correct? Right? Now here, if you see, all the pistils are, fused together. You can see the stigmas are fused together. On the other hand, when you look over here the carpels are free. You can see the carpels are free. They are not fused. Here they are fused. And when you look at this one, you can see over here, this is the hilum. So what is this hilum? The point of attachment of this funicle. This is the funicle, right? So the attachment of the funicle with the body of the ovule is going to be the hilum. Here you can see that this particular hilum, this particular cunicle is completely a part of the body of the ovule, right? So this is going to be called as a ridge. This is a ridge. Ridge matlab kya? Just like a connector, bridge ke jaisa hai, but it is completely inside it. So this cunicle is completely attached to the body of the ovule. This particular structure will be called as your raphe. So raphe kya hai beta? Raphe is a ridge, a bridge-like thing wherein both are completely fused. Hilum is this beta, this part. From here, the body of the ovule starts. From this line above is the funicle. From this line below is the body of the ovule. Okay, got it? Understood? So this line, this looks like a scar. It looks like a small, what do we say? एक ऐसा डार्कली कलर्ड पॉइंट दिखता है सीड्स के ऊपर. अगर आप सीड्स के ऊपर देखोगे तो भी आपको हाइलम दिखाई देगा. So it's a darkly spotted colored point रहता है छोटा सा. Got it? Yeah. So this is going to be the hilum. This is going to be the funicle. This entire one. Here it has got completely, inside the body of the ovule. So this is going to be the raphe. Hilum is also there in whom? Yeah. That is a different hilum. Kidney ka hilum is different, and this hilum is different. Okay. Well, so you understood this very clearly. How does it look like? What you want me to explain, beta? Hilum and chalaza. Hilum is this thing. This line that you can see over here is the hilum. Hilum is going to be the point at which the funicle is connected with the body of the ovule. So this is the body of the ovule. So above this is going to be the funicle. So funicle and body of the ovule attached to each other at a particular point. That point will be called as hilum. Okay. I haven't yet explained chalaza and micropyle. I'll talk about it. Wait. But have you all understood what is hilum and funicle? Any doubts with hilum and funicle in this diagram especially? Any difficulty? Understood? Hilum and funicle? Yeah. There's a window that is popping up and disturbing us now. I know. It disturbs me also. Okay. Now the window is gone. So we won't get disturbance. Well, the body of the ovule shows two ends. The basal end, often called as the chalazal end, and the upper end, generally called as the micropylar end. तो ये जो ओव्यूल है उसका एक बेस होता है और उसका अपेक्स होता है. तो ओव्यूल का जो बेस है ना बेटा, उस बेस को हम कहते हैं चलाजा, और जो एपेक्स है उसको हम कहते हैं माइक्रोपाइल. But ये micropyle कैसे बना? How this micropyle is going to be formed? I'll tell you that also. Okay. The main body of the ovule is covered with one or two envelopes called as integuments, which I've already told you. These leave a small opening at the top of the ovule, which will be called as the micropyle. That means, I hope you all can see my hands. Okay. That means the two envelopes are not going to be, completely fused. There's going to be a small gap. There's going to be a small gap, and this gap is going to be called as the micropyle. So how is the micropyle going to be created? Micropyle is created by a gap in between the two integuments covering the body of the ovule. Right? Got it? So थोड़ा सा जो एक point, open point रहता है ना, दोनों साइड से ये एनवेलप्स पूरी तरह से फ्यूज नहीं हो जाते बेटा, ये थोड़े से गैप रखते हैं. ये जो गैप है ना, उस गैप को हम कहते हैं माइक्रोपाइल. Well, the integuments encloses a large parenchymatous tissue called as nucellus. Now here is the point where you all will make a mistake. It's not nucleus, it is nucellus. What is it? nucellus. So what is nucellus? Body of the ovule is called as nucellus, which is a multicellular parenchymatous tissue. Fine. The residual part of the nucellus in the mature seed is called as perisperm. Residual part of nucellus. So rest of the nucellus will be eaten away, will go away. Where? Who will eat that? The embryo will eat that. The embryo will consume that for energy. कभी कभी बचा कुचा न्यूसेलस के सेल्स ना रह जाते हैं, जिसको हम कहते हैं पेरिस्पर्म. Okay, food laden means loaded with food. Where is that food laden? I didn't see that word food laden anywhere. Is there any word over here? Food laden? No. Anyways, loaded with food. What do you mean by food laden? Loaded with food. Okay. Towards the micropyle in the nucellus, a female gametophyte is situated, which is known as embryo sac. So now, see, beta, we have to be very precise with the female gametophyte. See, again, I'm telling you all, please listen to this very carefully. A flower. I know. Wait. What do you want me to explain? The third point? First, second, third. The two integuments. That's why I told you, if you can watch my hands, can you see my hands very clearly? If you can see, then you will understand this better. See, now I'll bring it closer. See, here now, these are the two integuments. So 2 means 1 plus 1. I have got only one, like to show. So 1 plus 1, 1 plus 1. They're going to be covering, they're going to be covering the nucellus completely, but they won't fuse like this way. There's going to be a gap. So this gap will be called as the micropyle. Understood? Okay. So the integuments leave an opening. They leave a gap. Okay. And these are going to be called as the micropyle. Like the covering of the seed. Yeah. Okay. Got it, beta? Now understood this, how it is? Perfectly? Okay. Then, towards the micropyle in the nucellus, a female gametophyte is there, which is going to be called as the embryo sac. Now, what is this embryo sac? I'll get the things reversed so that you will come to know. A flower having a pistil having style, stigma, ovary. Inside the ovary, there is going to be a nutritive tissue called as placenta. The placenta will have attachment of the ovules. They'll attach with the help of a stalk called as funicle. The point of the funicle, which is attached to the nucellus, is called as hilum. The body of the ovule is going to be made up of parenchymatous cells called as nucellus. The nucellus is going to be having two ends, chalazal end, micropylar end. Micropyle is a small pore that is left because the two integuments do not completely fuse with each other. The other end of the micropyle will be called as chalazal end. There is going to be the presence of an embryo sac inside this nucellar tissue. तो ये न्यूसेलस के अंदर एम्ब्रियो सैक नाम का एक स्ट्रक्चर फॉर्म होने वाला है. कैसे फॉर्म होने वाला है वो मैं आगे बताऊंगी. Of course, I'll tell you all. But is there any confusion with these basic things that I have explained just now? Anyone having any doubts? But just try to understand this, and if not, please tell me. I know you all are listening this for the first time probably, so you might have doubts, and if you have, it's okay. Ask me. In every nucellus, sac will be formed. Yes, better. Yes, that is the main role of the nucellus, to form the embryo sac. Nucellus is the body of the ovule. Body of the ovule का मतलब क्या है? देखिए, आप अगर इक्लेयर चॉकलेट खाते हो, हाँ, इक्लेयर चॉकलेट देखा है आपने? इक्लेयर चॉकलेट को देखा है? हाँ, तो eclair chocolate के ऊपर एक individual wrapping होता है कि नहीं? It's going to have a covering, right? So just like that covering, okay, that covering will be called as the integument. और melody chocolate ले लो, melody chocolate भी वैसे ही है, only on one side is going to have. So melody chocolate will be a better option, an example than eclair chocolate. So melody chocolate आपने देखी है, हाँ, melody chocolate के ऊपर wrapping होता है. It has got one pointed end, one end, right? So in the same way, your ovule looks like that, just like your melody chocolate. Okay, having that stalk called as the funicle. Correct? Got it? Now the point where it is going to be pinched, उसको ऐसे घुमा घुमा के ऐसा पतला कर देते हैं, that you can consider as the hilum. Fine. अब ये जो कवरिंग है ना मेलोडी के ऊपर पूरा होता है. अब आपको इमेजिन क्या करना है कि उसके बेस वाला जो कवरिंग है ना वो नहीं है. That is the thing that you just imagine, that it does not have the base wala covering. Then how it will look like? You can directly see the chocolate inside, right? So that point where you can directly see the chocolate inside na is going to be the micropile. Okay, and when you remove the wrapper, right? So when you remove the integuments or when you remove the wrapper, जो actual chocolate आपको दिखाई देगा ना, the actual chocolate which is going to be consumed by you all, right? That melody ka actual chocolate, that is going to be the nucellus. So now you could imagine what is it? Huh? Ab, if I tell you that ये जो nucellus है, it is multicellular. It is multicellular. Okay. In the same way, this melody chocolate is going to be made of many different ingredients. Okay. Now clear, beta? How does it appear like? Okay. Fine. Others, have you all understood? So only the one thing which you have to imagine is, what? The melody chocolate does not have a wrapper at the base, which is a bit difficult to, you won't buy such a chocolate, I know, which is not wrapped completely. But for the sake of understanding this concept, it becomes very simple to understand this. Perisperm, yeah, see what happens is, beta, when the embryo is developing, it's going to eat away all the cells which are coming in its way. Huh? So let it be the nucellular cells or let it be the embryo sac के cells, it's going to eat away. खाता जाता है वो, है ना? और जब वो खाता है ना, तो जनरली न्यूसेलस के सारे सेल्स वो खा के खत्म करता है, राइट? बट कभी कभार है ना, few cells will remain. If those few cells remain not eaten, they are not eaten by the developing embryo, अगर वो रहते हैं, बचते हैं, then those बचे कुछ cells को हम कहते हैं perisperm. Okay, got it? Second point, the main body of the ovule, the chocolate is covered by a wrapper. So the main body of the ovule, that is the nucellus, is covered by 2 or 1 envelopes, which are going to be called as integuments. Okay. Raphe is, okay, I'll tell you what is raphe now. With an example, embryos form where in the ovule, I'll tell you. It's a complete process, how and where it is going to be formed, I'll tell you. So, for example, raphe is going to be, there's going to be the chocolate ka wrapper that I told you just now, right? Which is going to have that one side wala thing. Okay. Now imagine that the chocolate is going to be, it's melted chocolate and it comes out and covers the entire plastic, leaving only the apex. How will it look like? वो मेलोडी चॉकलेट इमेजिन कर लो पूरा पिघल गया और पिघल के उसने पूरे रैपर के ऊपर आके एक लेयर बनाया, तो कैसे दिखेगा, है ना? So now where is that wrapper? Wrapper is now sandwiched between whom? The chocolate. Okay, then that is going to be the wrapper, something like that. So the that filament, which is actually the funicle, will enter directly into the nucellus and will be a part of it. Then how will it look like? Na? Something like that. Okay, now got it? I understood this? Well, या फिर यू कैन टेक अ बाउल, एक कटोरी ले लो, हाँ, और उस कटोरी के साथ एक रॉड, उसके साथ स्टील का रॉड ऐसे अटैच कर लो कि वो है ना, वो सर्विंग के स्पून बन जाए. हाउ इट विल लुक लाइक? समथिंग लाइक दैट. So it's going to be a part of that entire, it's going to be a part of that entire ovule. It's not separate now. Then it will be called as racket. ठीक है? Now fine. Okay. Anyone else having any difficulty till here? Whatever we have done. Simple. Now making the things okay now. Going in your head. Well, can I move ahead, beta? Can I go ahead? Any points of your earlier point? कुछ difficulty है? जाऊँ आगे? Fine. Well, so this diagram so I had already explained. So this is what I'm saying. See, this is like a serving spoon हो गया ये अभी, ये देखो, ये इसके अंदर ही पूरा आ गया ना, तो it's going to act like a serving spoon. Now look over here, beta. This is the diagram which I was talking about. So this is going to be the funicle. Can you see this? 1 and 2. These are the two integuments. This is the outer integument. This is the inner integument. This is the nucellus. Can you see this? Nucellus. Right? Nucellus के अंदर क्या है? Embryo sac. So this is the nucellus. Nucellus के अंदर embryo sac. Nucellus is covered by two integuments, inner and outer. The inner and outer integuments do not fuse with each other. They leave a small pore. This pore is going to be called as micropyle. And this end is called as micropylar end of the ovule. The end opposite to the micropylar is going to be the chalazal end. So you can see over here the chalazal pore. So now you understood how does this entire assembly look like with the diagrams. Clear? Okay. So here they are saying a dissected flower of hibiscus showing pistil. Look over here. This is the hibiscus flower का dissection किया हुआ है. This is multicarpellary, means many carpels. Syncarpous means fused together, as seen in papaver. Okay, papaver जो होता है ना, that is your poppy seeds. खसखस, खसखस पता है? poppy seeds, उसका flower है. ठीक है? This is going to be the multicarpellary apocarpus, means without fusion. Here the carpels are not fused. Here the carpels are fused. Here the carpels are free. Okay, so free carpels, as seen in Michelia. Michelia is in Marathi we call it safa. You know what is safa? Okay, so that typical, like very nice. उसका परफ्यूम बहुत मस्त होता है, उसका ऑर्डर बहुत सही रहता है. Yeah, we wear it in the hair also. We offer it to the, yeah, orangish yellowish color. Yes, saffron. Yes, yes. Yellowish orangish type shade होती है उसकी. So nowadays it's found in the market. अभी उसका season चल रहा है फिलहाल. You can, if you go to the flower market now, you can see that. So that is going to be the apocarpus. That is going to be your Michelia. And here it's a diagrammatic view of typical anatropous ovule. Now, what is anatropous ovule? I'll explain. Don't worry about the name, but at least you will have understood the structure very clearly. Micropyle is the pointed top part of the ovule. No, beta, this is micropyle is this depression. ये gap जो दिखाई दे रही है ना, जहाँ पर integuments are not fusing. This gap is going to be the micropyle. Chalaza will not have a hole. It's an entire thing. Chalaza does not have a hole. It's just the opposite side of the micropyle. Okay. Micropyle is just the name of this gap. Yes, yes, yes. Perfect. This is the gap. This gap, that pore or the opening will be called as the micropyle. हाँ, चंपा या चंपाइन हिंदी कहते हैं उसको अपने मिशेलिया से. Okay. Chalaza has no function. Practically no function, but eventually you'll see that why it is demarked as chalaza. Not functionally, but because of some reason we have to demarcate is as the chalaza line and the microphylla line. Okay, that you will come to. That will come to. So you understood the meaning of these? Syncarpus, fused. Carpels are fused. Apocarpus, free. They are not fused. When the carpels are fused, we say syncarpus. When the carpels are free, we call it as apocarpus. Huh? Okay. Then let us talk about the types of integuments. So we have got something called as unitegmic. As the name says, uni means one. So ovule with a single integument. Example, sympatalus or gamopetalous dicotyledons. Forget about that. What is sympatalus or gamopetalous? Zarurat nahi hai. Just remember, unitegmic means having a single integument. Means only one covering. Outer and inner ऐसा कुछ नहीं होता है, एक ही covering होता है. Okay. Bitegmic, ovule with two integuments. Okay. Outer and inner. Okay. No need of remembering these things. Huh? That's extra. That's okay. Third is important. Okay. Third is aril. Now, what is an aril? Please remember, this is a collar-like outgrowth from the base of the ovule and forms a third integument. Okay. So normally one or two integuments is very common. Two integuments is maximally found. Now it's going to have a third integument. Then the third integument will be called as what? Aril. And do you know that aril is eaten by us in case of lychee? Lychee खाते हो? Lychee खाते हो कि नहीं? हाँ? हाँ, lychee मस्त होता है ना? Yeah, even I like lychee. तो जो लीची हम खाते हैं ना बेटा, that fleshy part of the lychee that we eat is actually the third integument that we are eating. Oh my God, you might have never thought of it, na? So what do we eat? We eat the third integument. We don't consume, so जिसको हम fruit के नाम से खाते हैं, what is it? It is the third integument which becomes fleshy. Okay, and we eat that aril. नहीं, apricot is the seed के अंदर का endosperm that we eat. I'll talk about it. It's a very interesting case. Okay. Well, so aril समझा. In case of lychee, nutmeg, जायफल. मराठी we call it जायफल. पता है आपको? वो nutmeg ज़्यादा खाने से नींद आ जाती है, पता है? You should not eat a lot of nutmeg. If you put, generally we put it in coffee or like generally in whatever sweets we make, na, we add a little bit of nutmeg powder to it. Jaifal ka powder, yeah, in kheer also. Yeah. Exactly, Vaishnavi. Perfect, bitter. So that is the thing, like you should not eat a lot of it. So that is going to be the third integument. Got it? What is the third integument? Fine. It has got the sedative powers are there in them. The chemicals are sedative. They give you drowsiness. Okay. Then the fourth is caruncle. This is also an important point, beta. Caruncle. It is formed as the outgrowth of the outer integument at the micropylar region. So what will happen? The micropyle will not be seen. So there won't be any micropyle. The outer integument is completely fused. Okay. So you cannot get that micropyle over there. You cannot see the micropyle over there. That's going to be called as caruncle. So what is caruncle? The outgrowth of the outer integument, which will completely fuse, not having a gap in between, not having a pore in between. So there won't be any micropyle present in them. That's called as a caruncle. Castor. Castor seeds. Have you all seen castor seeds? I don't know if you have seen castor seeds. You know castor oil. But I am not sure if you have seen castor seeds. Deखे hai? Castor. If you have seen, then you might know what is a caruncle. They completely not seen, I know. They completely fuse and they don't leave a micropylar end. Okay. Ategmic means without integuments. So in some parasitic plants, like Loranthus, Viscum, Santalum, there is no integument. Such an ovule is called as ategmic. They don't have any integuments because they are parasitic plants. So parasites hai to they have to be always dependent on the host for the nourishment and everything. So why do they need care and why do they need protection? So they won't have. Fine. So unitegmic, bitegmic, ategmic. Uni means one integument, bi means two integuments, ategmic means no integument. Aryl is the third integument. Caruncle is the outer integument which is completely fused. Yeah, that's what I said. Caruncle is the outer integument which is going to be completely fused, not leaving a pore, not having a gap. Okay, so you will not be able to see the micropyle in case of caruncles. Is it okay? Understood the types of integuments? Any difficulty? Any difficulty, beta? All of them, all five of them. Just review once and let me know if you've got any problem. देखो, no need of remembering the examples. That's okay. That's never going to be asked. Fifth one will not have any covering. No covering at all. Okay. Micropyle gap is filled. Yeah, gap is filled in the sense there is no gap only. Na. It's completely meeting. पूरा ऐसा हो गया वो यहाँ से. Okay. Well, so that's about the types of integuments. Now let us see the different types of ovules. There are different types in different types of angiosperms depending upon the position. Okay, depending upon the position, we have got different types of ovules. The first is orthotropous ovule, also called as atropous. Now, my dears, this is important. It's not given in NCERT, and they're going to ask you questions for match the following in this. So integuments includes the fleshy part. Not necessary. Now, moong ka chilka is thin. है ना? Peanut ka chilka is thin. Okay. It depends. It depends on the species. Well, so let us talk about these. But instead of first talking about all these things, I'll explain with the help of the examples. It will be easy for you. Look at this. This is the orthotropous ovule. Here you can see this is the micropyle. This is the chalaza. This is the funicle. Correct? Micropyle, chalaza, funicle. When all three of them lie in the same line, in the same plane, we call this type of ovule as orthotropous ovule. Which one? Micropyle, chalaza, and funicle all lie in the same plane. We call it as orthotropous type of ovule. Understood what is orthotropous? So now look at the description. The micropyle, chalaza, and funicle are in a straight line. Correct now? Understood? Any difficulty with this? Fine. Yeah. Then the next one. Example is beetle, piper, polygonum. No need of remembering. This is just for making you all understand. Okay. Beetle पता है ना beetle? Beetle leaf. पान खाते हैं ना जिसका पान that you eat. That is going to have orthotropous ovule. Piper is kali mirch. Piper is kali mirch. Black pepper. Polygonum is not eaten or it's not consumed. Anatropous. Let us talk about the second one. What is anatropous? Look at here. So this is going to be the funicle. Can you see? This is the funicle. Chalaza is upper and micropyle is niche. So don't you think ये जो orthotropous है उसने एक पूरा U-turn ले लिया 180 डिग्री टर्न लिया कि नहीं उसने हाँ ये देखो ये ऐसा है अपना orthotropous इसने क्या किया 180 डिग्री टर्न लिया so because of that what happened the funicle look over here the funicle and the micropyle are lying close to each other and the chalaza has gone above now चला जा ऊपर चला गया, माइक्रोपाइल नीचे आ गया, और माइक्रोपाइल और फ्यूनिकल बहुत क्लोज आ गए एक दूसरे के. This type of ovule will be called as anatropous ovule. And let me tell you, 82% of the angiosperms have got anatropous ovule. Okay, so maximum angiosperms are going to have anatropous ovule. Okay, so see, the body of the ovule is completely inverted. Turn is at 180 degrees angle, so that micropyle and hilum come to lie very close to each other, or micropyle and funicle come to lie very close to each other. See, 82% of the angiospermic families are going to have anatropous ovule. So even we are going to talk about anatropous ovule only आगे जाके. जब हम ओव्यूल के बारे में पढ़ेंगे ना आगे, तो हम सिर्फ एनाट्रोपस ओव्यूल के बारे में पढ़ेंगे. We are not going to talk about the others, but you should know what are the others. Hemianatropous. Look at this. 90 degrees. Can you see? Hemianatropous. So the funicle and the micropyle are at 90 degrees from each other. And chalaza bhi idhar 90 degrees pe aa gaya hai, dekho. Correct? So look at this. Ovule turns at 90 degree angle upon the funicle, or the body of the ovule is at right angle to the funicle. Ya na? Look at it. It's at right angle, na? So this is going to be called as hemitropous or hemianatropous. Fine. Fourth is compylotropous. Ovule is circled more or less at right angle to the funicle. More or less means it's not going to be 90. It's going to be between 90 and 180. हाँ, थोड़ा बहुत slanting position में है. And micropyle is bent downward slightly. Look over here, compylotropous. ये देखो. It's not here ना. ये देखो, look at the difference between the hemianatropous and the compylotropous. Look at this. It's going to be bent downwards, but it is not completely 180 degree also. So such type of ovule is going to be called as compylotropous. Fine. Here you can see amphitropous. Amphitropous. The curvature of the ovule is more, and the embryo sac becomes curved like horseshoe. Look at this embryo sac का shape, है ना? कैसे हो गया देखो, horseshoe के जैसा हो गया है. करेक्ट. तो हमेशा वो लोग कहाँ पर आपको पकड़ते हैं पता है? इन दोनों में. Anatropous and amphitropous looks very much similar, but you have to detect based on what? The shape of the embryo sac. So this is the embryo sac which is oval. This is the embryo sac which is horseshoe shape. ठीक है? And the last is circinotropous, which is 360 degrees turn. So it's just 360 degrees. So orthotropous becomes anatropous, again becomes orthotropous. That is called as circinotropous. Look over here. The ovule is initially orthotropous, but becomes anatropous due to unilateral growth of the funicle. The growth continues till the ovule once again becomes orthotropous. वैसा पूरा एक U-turn लेके आ गया वापस. As a result, funicle completely surrounds the body of the ovule. Look over here. See, orthotropous था, it became anatropous and again became orthotropous, but the funicle is completely covering the entire ovule. So this is your circinotropous. Please remember these names. Very simple. Shortcut there you. Ortho, all in one row. Ana, 180 degree. Hemi, 90 degree. Compylo, somewhat irregular. Amphi, horseshoe type of the embryo sac. Circino, 360 degrees. Simple now. Got it now? This is the previous slide. Just check it out if anyone has got any difficulty with understanding of these things. Anyone having any problem with understanding of these, beta? What are the types of ovules? So we have seen the types of integuments. We have seen the types of ovules. Both we have seen in detail, and very important points we have discussed. Compylotropous. Crucifera is going to be in the compylotropous. I gave a shortcut. What is the shortcut? Orthotropous, all in one row. Who all? Micropyle, funicle, and the body of the ovule, right? Anatropous 180-degree turn, hemianatropous 90-degree turn, compylotropous a bit irregular, more than 90, less than 180, amphi, horseshoe shape wala embryo sac, and silsilo 360 degrees. Yeah, exactly. In the beginning, they all are orthotropous. Eventually, they change their shape depending upon the development of the funicle. How the funicle is going to grow, accordingly, they have to turn. Right? Got it? Clear about it? I'll go ahead. Sequence? No. Sequence could be anything the way you can remember. But we are going to talk about the anatropous ovule only in general. Sequence, whatever, the way you want to. Okay, now we come to megasporogenesis. How we had talked about microsporogenesis, how we had done in detail about the formation of the microspores. In the same way, we're going to do the formation of the megaspore. Okay, देखो. The ovule or the integument or the megasporangium develop as a small protuberance of the placental tissue. Protuberance का मतलब होता है outgrowth. What do you mean by the word protuberance? Outgrowth, छोटा सा, एकदम से हल्का सा. Okay, so the ovule or the megasporangium develops as a small protuberance of the placental tissue. So the placental tissue is going to have small outgrowths coming out of it. That is going to be the beginning of the formation of the megaspore. In the very young ovule, a single hypodermal cell is differentiated as archaesporal cell. Do you recollect the language? Same language was there in microsporogenesis also. In the young ovule, here, a single hypodermal cell. So epidermal cell is always going to be there. Uske niche wala hypodermal cell is differentiated into archaeosporium cell. Same we had seen in microspore also. Correct? The archaeosporial cell may directly function as megaspore mother cell, or it may divide periclinally to form outer parietal cell and inner sporogenous cell. So same na, I have told you outer is always parietal. In case of animals ho ya plants ho, outer is parietal. Inner is visceral in case of animals and sporogenous in case of plants. अब periclinally का मतलब क्या होता है वो बताती हूँ मैं. Okay, just pay attention to this. This is going to be repeated again and again. Periclinal and anticlinal, ये दो words important है बेटा. So please understand what are these two words. I'll just try to draw the diagram so that you will understand the things very clearly. Please pay attention and please make a note of it in your books. Nowhere in the books, any reference book also, they have not mentioned what is the meaning of periclinal and anticlinal. So kindly note these things, okay? One. Yeah. So I'm telling you what is periclinal and what is anticlinal. So you can see these boxes, right? Assume these boxes to be the cells. Assume the boxes to be the cells. Okay. Now, when a cell will divide, periclinally, I'll first tell you what is periclinally. So what do you mean by this word? Periclinal axis, periclinal. So don't you think this will lead to the formation of two lambe? Array, sorry, I only forgot कि मैंने कौन सा select किया. This will give rise to what beta? Which type of cells will it give rise to? ऐसे दो लंबे cells को ये जन्म देगा कि नहीं after division, right? So this will give rise to two these type of cells, and this will give rise to two these type of cells. but both cells how are they are along the axis. So this type of cell division को हम कहते हैं periclinal. What we call it as periclinal. Huh? Now let us see what is anticlinal. ये देखो, ये सेल और ये सेल सेम शेप के हैं, ये सेल और ये सेल सेम शेप के हैं, but look at the difference now. I'm making anticlinal. Can you see? So because of anticlinal type of division, the shape of the cell has changed, right or not? Now the shape is square rather than becoming elongated. So this type of cell division is going to be called as anticlinal. Got it? Understood what is periclinal and anticlinal? Periclinal cell division, and this is going to be no, longitudinal is periclinal nahi hai beta. See, here it's going to be, like this way. So here it seems to be transverse. So when we say periclinal, it has to be along the axis, and when we say anticlinal, it is against the axis. Okay, got it? Understood very nicely. Any difficulty in understanding this? Now see, periclinal division will give rise to, increase in the height, and anticlinal will give rise to increase in the width. So if both are going to take place, overall structure will become big. Clear? Have you all drawn these things? I want you all to draw this because it's going to be repeated again and again in many chapters. So again, I don't want a question that means what is periclinal and what is anticlinal. So kindly note down these things. Come on, quickly. I'll give you 2 minutes. गुड मॉर्निंग है कि नहीं सबका? हाँ, कि फेस्टिवल्स में इतना मीठा खाया कि अभी नींद आ रही है? इज इट लाइक दैट वे? बहुत ज्यादा मीठा खा गया तो नहीं है 4-5 दिन में? उठा उठा कर जितना भी लड्डू है, मोदक है, जो भी है, मोदक, लड्डू, और क्या खाया? हाँ, ज्यादा खाओगे तो फिर यू विल बिकम मोर लेजी. हाँ, हाँ, पेड़ा, वही तो, लड्डू, मोदक, पेड़ा, मस्त. अरे वाह, वेरी नाइस. मैसूर पाक भी खाया? नाइस, आई लाइक दैट. Yeah, Janvi, I will. I'm just waiting for everyone to copy down, beta. I will. I read your. चलो डन बेटा, all of you all have done. ड्रॉ किया, ये लिखा. Yeah. So, what is periclinal? Peri is along the axis. The cell division will take place along the axis, and anticlinal, the cell division will take place against the axis. So this particular division, see, along the axis. This is also along the axis. ये एक्सिस लॉन्जिट्यूडनल है लेकिन ट्रांसवर्सली कट हो गया. ये देखो, ये लॉन्जिट्यूडनल है, ये ट्रांसवर्सली कट हो गया. हाँ, सो इट इज अगेंस्ट. सो अलोंग द एक्सिस, अगेंस्ट द एक्सिस. ठीक है? गॉट इट बेटा? क्लियर? Great. So we'll move ahead and stop the share of this thing, and we'll go ahead with the formation wala part. हाँ, या, yes, Aditi. Yes. Okay, now let's go to the formation of the megaspore. As I said, this is a very important point. So that is what I was trying to tell you periclinally. So it may divide periclinally to form an outer parietal layer. So you now can imagine that outer layer would be the parietal and inner would be the sporogenous layer. If directly that one single cell will act as MMC, means it will undergo meiosis, we call it as tenuinucellate ovule. And if it is going to undergo mitosis with making of parietal cells and sporogenous cell, we call it as crassinucellate ovule. Okay, so tenuinucellate में क्या होता है? एक सेल है, माइटोसिस अंडरगो नहीं करेगा, सीधा मियोसिस अंडरगो करेगा, तो वी कॉल इट एज tenuinucellate. एक सेल है, वो पेरिक्लिनल एंटीक्लिनल डिविजन्स करते करते पेरिक्लिनल डिविजन्स करते 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 इट विल मेक टू लेयर्स, outer parietal and inner sporogenous, and such type of particular structure of the megaspore will be called as crassinucellate ovule. Okay, remember the words beta tenuinucellate and crassinucellate. I know it is out of syllabus wala content, but these things have been asked in the previous year's question papers, so I have deliberately included all these things. Okay, the sporogenous cell directly behaves as megaspore mother cell, or we call it as megasporocyte. Okay, so outer layer will not be fertile, but the sporogenous layer will be fertile, means it can undergo meiosis. Okay. Then the diploid megaspore mother cell enlarges in size and divides by meiosis to form a linear tetrad of 4 haploid megaspores. You already know protuberance means outgrowth. Protuberance means outgrowth. Okay, so diploid mother cell, नाम से ही पता चलेगा. Mother cell means it's going to undergo what? Meiosis. And after undergoing meiosis, what will happen? It's going to form a tetrad of megaspore. So 4 haploid megaspores arranged in one row. We have seen, you remember? Tetrahedral tetrad, decussate tetrad, linear tetrad. You remember we have seen all these in the microspore formation. So here what will happen? There would be 4 megaspores formed in a row, right? So the mother cell, megaspore mother cell, after undergoing meiosis, forms 4 haploid cells in one row. These are going to be called as a tetrad. Occasionally T-shape and inverted T-shaped tetrads are also formed, but we are not bothered about it. Megaspore is the first cell of female gametophyte. This is a very favorite question, that out of the following, which represents the first cell of female gametophyte? So it's the megaspore which will represent the first cell of female gametophyte. Of the linear tetrad, 3 megaspores towards the micropyle degenerate. The lowermost, that is the chalazar microspore, enlarges and remains functional. This one will be called as a functional megaspore. It later produces an embryo sac. Now, this is the most important point, beta. I'll show you with the diagram. Don't worry. See. So this is going to be the parietal cells. Outside the parietal cells, that is the nucellus. Here it's going to be the megaspore mother cell. The sporogenous cell का ये megaspore mother cell. This megaspore mother cell, यहाँ पर है micropylar end, यहाँ पर है chalazal end, will undergo first meiotic division to form 2 cells. That is called as megaspore dyad. Meiosis 2 will give rise to 4 haploid cells, that is called as megaspore tetrad, which is in one row. Can you see? Right? Now, out of these, 1, 2, and 3, all 3 will degenerate. All 3 will degenerate. Who will survive? Only one will survive. The one towards the chalazal end will survive. Did you understand what they are trying to tell you? अब समझ में आया ये कैसे है? I know, फिर भी समझना मुश्किल है, तो मैं ड्रॉ करके दिखाती हूँ तुम्हें. Okay, I'll draw it for you. Wait. Just now understand this very nicely, beta. For better nourishment, they are going to degenerate because they have to give better nourishment. Okay. Now I'll just talk about this. एक एक करके मैं आपको ना सिस्टमैटिकली बताऊंगी ना, तो ये आपको एकदम क्लियर हो जाएगा. डोंट वरी, नथिंग टू वरी अबाउट. ओके. So this is going to be the stigma. Okay. This is going to be the style. This is going to be the ovary. Okay. नीचे अपना थैलेमस है. बराबर. Stigma, style, and ovary. Inside the ovary, here there is going to be the placenta. ये क्या है? ये जो टिश्यू है, ये कौन सा टिश्यू है बेटा? प्लेसेंटा. प्लेसेंटा से अटैच, ये देखो मैं इधर बना रही हूँ, ये फ्यूनिकल और ये ओव्यूल. राइट. दिस इज द फ्यूनिकल. This is going to be the ovule. You can have one ovule or many ovules, that doesn't matter. Okay. Now, ये जो एक सिंगल ओव्यूल है ना, इसको मैं जूम कर रही हूँ. आई एम एनलार्जिंग दिस सिंगल ओव्यूल वाला पार्ट. ये वाले पार्ट को मैं जूम करती हूँ. So if I zoom this, what will I get? If I zoom this, I'll get something like this way. Right? I'll get something like this way. And I'll be having मैं एनाट्रोपस ओव्यूल ड्रॉ कर रही हूँ बेटा, ताकि तुम्हें ना कंफ्यूजन पहले से ही ना हो. तो ये एनाट्रोपस ओव्यूल है, इसलिए मैं ऊपर की तरफ माइक्रोपाइल लेके जा रही हूँ. दिस इज वन ऑफ द इंटेगुमेंट्स एंड दिस इज गोइंग टू बी द सेकेंड इंटेगुमेंट. करेक्ट. हमने अभी अभी देखा बाइटेगमिक ओव्यूल है, तो ये दूसरा इंटेगुमेंट. सो दिस इज गोइंग टू बी माय micropyle. This is going to be my chalaza. इधर तक ओके. ठीक है. अब इसके अंदर, inside this, I'm drawing multicellular structure, that is prassinucleate ovule बना रही हूँ मैं यहाँ पर. मल्टीसेल्यूलर ऐसे मैंने एक ओव्यूल यहाँ पर बनाया है. राइट. It's going to have its own nucleus. Every cell will have its own nucleus. ये देखो. Okay. Got this? How does it look like? See this. Well. So this is the way it looks like. Now ये जो black color के cells मैंने बनाए हैं ना, these cells are going to be your nucellus. What is it? Nucellus. ये जो black cells है, what are these? The nucellus or the body of the ovule, as I always say. Now, one of the cells near the micropylar end, suppose this cell at the micropylar end, decides to act as megaspore mother cell. Okay? तो अब मैं इसको zoom कर रही हूँ. समझा मैं किसको zoom कर रही हूँ? Now I'm trying to zoom whom? I'm trying to zoom the megaspore mother cell. Okay? So this is the megaspore mother cell, जिसको मैंने zoom किया. Okay? What it will undergo? It will undergo meiosis to form a tetrad. Can you see? This is the way a tetrad is formed. Right? It's going to have its own nucleus. Huh? It's going to have its own nucleus. Okay? अब यहाँ पर देखो बेटा, ये कौन सा एंड है इसका? ये माइक्रोपाइलर एंड है. ये कौन सा है? दिस इज द chalazal end. Correct. Now what's going to happen is, ये देख, out of these four, the three towards the micropyle will degenerate. तो ये गया, ये भी गया और ये भी गया. So who survived बेटा? Who survived? हाँ, in Anatropous, yes, beta, you are right. You're perfectly right. Siddhika, you're perfectly right that in Anatropous it should be down. Agreed. But this is the diagram that they have given in your textbook, na. So I'm drawing this. Okay. So that is the reason. Okay. Otherwise, we are going to talk about the Anatropous only. But there's always a confusion because the textbook says something different. So I'm going according to your NCERT textbook. So textbook is showing this diagram, right? So who is surviving now? Can you tell me? Only this one towards the chalazal end is surviving. Correct? So you understood how the things are progressing. Now we have to place this chalazal wala over here. So ये जो chalazal end वाला है वो actually इधर वाला एक बचा कुचा एक point है. तो ये जो chalazal end वाला जो बचा हुआ है ना, this cell, उसको अब मैं zoom करती हूँ. Okay? Now I will zoom this one. Okay? Now I am zooming this one. So this is going to be somewhat like this. Okay now? So this one is going to be this. This megaspore too. Yeah, every megaspore will have this. Huh? Everyone will have. Now can you see this? How I've done it? And inside this, you know, we have got one nucleus. There is one nucleus over here that you can see very clearly at the center. This one. Now this one will be called as, what we call it as? Functional megaspore. Did you understand what is functional megaspore? Any doubts with the word functional megaspore? So the spore which survives, the spore which sustains, the spore which remains is going to be called as functional megaspore. Okay. Now what will happen in this functional megaspore? Let us understand. Okay. So I go ahead with the explanation. So this functional megaspore will undergo what it will undergo, beta? First mitosis. So first mitosis will lead to what? Do you know? First mitosis will lead to what? Two cells. But you know what will happen? The mitosis is actually going to give rise to only two nuclei. It's going to give rise to only two nuclei because the mitosis is going to only take place in the nucleus and not in the cytoplasm. Understood what has happened? Then it will give rise to, yeah. Then there will be, just like in fungus, how we have seen the dikaryophase, right? Same way. Yes. Very good, Siddhika. Very good answer. Very nice. Yes. Then it will undergo what they will undergo, see? Second mitosis, the same way as it had happened earlier. So what do you think? How many nuclei would be now made? Can you all tell me? How many nuclei will be made now? Very nice answer. Perfect answer. So look, even I'm increasing the size of this functional megaspore. Beta. So now it is going to have 1, 2, 3, and 4 nuclei. Do you agree? Okay. Now we'll go with the next one. Next क्या होता है देखते हैं. Let us see what's going to happen next. So this will undergo see, third mitosis. Ab bolo beta. Now what would be the condition? Now what would be the condition? Very nice. 8 nuclei. Very correct. So you see, I'm increasing the size of the megaspora. So 1, 2, 3, 4, 5, 6, 7, and 8. Can you all see? 8 nuclei being formed. So just imagine like how the things are progressing now. So what happened first, beta? There was a small protuberance, a small outgrowth on the placental tissue. Okay, that outgrowth enlarged. Either it act directly as MMC and underwent meiosis, या फिर it underwent mitosis to form sporogenous tissue inside, and outside there is another layer of the visceral tissue. Okay. Now this sporogenous, parietal tissue. Now this sporogenous tissue का एक cell, that is the nucellus का एक cell, it acted like megaspore mother cell. It underwent meiosis to form 4 haploids, that is linear tetrad. Out of the 4, the 3 towards the micropylar end degenerated. Only 1 towards the chalazal end sustained. The one towards the chalazal end then underwent first mitosis to form 2 nucleated structure, second mitosis to form 4 nucleated structure, and third mitosis to form 8 nucleated structure. Okay, so how many times the mitosis has taken place? Can you tell me? How many times the mitosis has taken place? 3 times. 3 times, exactly. Okay, now if you look at this particular structure that I've drawn, you can see that I have done a typical arrangement of that nuclei in a very specific manner. The reason is after, only 3 times. No, only 3 times. Okay, so it is going to give rise to what? It's going to give rise to 8 nuclei. इधर तक का कहानी समझा क्या बेटा? इधर तक किसी को कोई प्रॉब्लम है समझने में? Carpel, made up of stigma, style, and ovary. Ovary has a placental tissue. Outgrowth of the placental tissue will be making an ovule. That ovule could be single celled. If it is single celled, directly it will give rise to 4 megaspores. If it is undergoing mitosis, that is periclinal division, it will give rise to a group of cells called as sporogenous cells, called as nucellus. One of the cells of the nucellus near the micropylar end will act as MMC. It will undergo meiosis to form 4 haploids. The 4 haploids will be called as linear tetrads. Out of the 4, the 3 towards the micropylar end will get degenerated. Only 1 towards the chalazal end will survive. The one which is surviving is going to be called as functional megaspore, which will undergo 3 consecutive mitoses to form 8 nucleated structure. Any difficulty? Yeah, Sadhika, very correct. 8 nuclei, 3 up, 3 down, 2 at the center. Yes. All of you all have understood this. One more time, I'm repeating it. Please pay attention. Gynoecium is made of carpel. Carpel is made up of stigma, style, and ovary. Ovary contains placental tissue. Outgrowth of the placental tissue is going to make the ovule. Ovule could be single celled, or if it undergoes mitotic divisions or the periclinal mitotic divisions, it will become multi-celled. Multi-celled ovule will have nucellus, that is the sporogenous tissue का पूरा layer is called as nucellus. One of the cells of the nucellus near the micropylar end, please remember, near the micropylar end will undergo meiosis to form four haploids called as linear tetrads. Linear because they are in one row, in one line. Out of the four haploids formed, three would degenerate, which are going to be towards the micropylar end. The chalazal end megaspore will survive, which is going to be called as functional megaspore. The functional megaspore will undergo mitotic division 3 times only with the nucleus, that is only karyogamy will take place, which will lead to 8 nucleated structure. Any problem? Any difficulty? Come on, tell me better quickly. है ना? You had it ना? Yeah, some schools, yeah, some syllabus ना, they have this earlier. But this is a bit more in detail ना? चलो चलो पूछो, कुछ डाउट है तो पूछो यहाँ तक मुझे, कुछ भी नहीं समझाओ तो प्लीज आस्क मी, बिकॉज इसके आगे बहुत इंपॉर्टेंट चीज है मैं आपको बताने वाली हूँ. Perfectly understood. Can I ask fill in the blanks? Would you fill it up? Would you fill it up? I know, that is why I said I'll draw the diagram for you all. It will become more easy to understand. Okay, chalo. Well, gynoecium is made up of dash. फटाफट आंसर लिखते जाओ. Very good. Yes. Very nice. Very nice. Very good. Yeah, it's made up of carpel, beta. Yeah. Carpel is made up of dash, dash, and dash. Very good. Yes, yes, yes. Very nice. Correct. All correct answer. Very nice, beta. Stigma, style, and ovary. Correct. Ovary has a nutritive tissue called as dash. Good. Good. Yes. Very good. Very good. Correct answers. All are giving correct answers. Oh my God. Very nice. Very nice. Yeah. So this placenta is going to be the nutritive tissue. And outgrowth of this placental tissue will give rise to dash. Very good. Very good. It's going to give rise to, very nice, beta. Very correct answers. It's going to give rise to ovule. Very nice. The ovule could be single celled or it could be multi-celled. If it is multi-celled, then the tissue will be called as dash. Very good, Zul. First to answer. Very nice. Very nice. The tissue will be called as what? यहाँ पर बच्चे कंफ्यूज होने के चांसेस है whether it will be called as sporogenous or nucellus ना? Yeah, it's going to be called as sporogenous tissue. Yes, sporogenous tissue. And all the tissue together will make a structure called as body of ovule. That's nothing but that's the nucellus. Very correct. That is the nucellus. Perfect, beta. So individual cell is sporogenous cell, and the tissue that is going to be formed of the sporogenous cell will be called as the nucellus or body of the ovule. Okay. One of the cells of the nucellus near the dash end act as MMC. Very nice. Come on. Towards which end will act as MMC? No, it's not chalazal end. Which end will act as MMC? Look at this diagram. Look at this diagram that is there in front of you. Look over here, beta. Here is the micropyle, na? So near the micropylar end will act as MMC. Correct. Okay. Now, this MMC will undergo meiosis to give rise to dash tetrad. Which type of a tetrad? Excellent. Very correct answer. Very correct answer. Someone others give? Megaspore tetrad, in which form? How are they in one row? Na? So what we call them as? What we call them as? Linear tetrad. नहीं बेटा, orthotropous is the position of the ovule. Ovule के अंदर की ये सारी चीजें हो रही है बेटा. ओव्यूल का पोजीशन कैसा भी हो, उसके अंदर ये सारी चीजें हो रही है ना। तो ये जो है ये ओव्यूल का पोजीशन है, ये जो मैंने ओव्यूल का पोजीशन, ये देखो यहाँ पर, कैन यू सी, ये मेरा ओव्यूल का पोजीशन है, which is somewhat hemianatropous that I have grown, okay, but उसके अंदर ये सारी चीजें हो रही है, right, right, so it's a linear tetra, correct. So you can see all these 4 in one row, 1 2 3 4, linear tetra. Out of the 4 haploid cells that have been formed, 3 will degenerate while 1 will survive. The one which survived lies towards the dash end. Now you all are right with the answer. Yes. Correct, correct, correct. With the chalazal end. Very nice, very nice. The one which is going to survive near the chalazal end will be called as dash. It will be called as dash. Very good answers, very good answers, perfect answers. Yes, yes, perfect. It's going to be called as, you have written already, functional megaspore. Correct? Functional megaspore will undergo dash divisions to form 8 nuclei. How many mitotic divisions? 3 mitotic divisions. Very good. To form the 8 nuclei. Very good, beta. So anyone having any difficulty in understanding यहाँ तक की कहानी? कोई difficulty है? किसी को भी नहीं समझा है, ऐसा कुछ है? अभी क्लियर है ना? बिकॉज यही बहुत इंपॉर्टेंट है रे, ये समझा तो आगे का बहुत सिंपल हो जाएगा, बट ये इसमें कंफ्यूजन रहेगा ना तो सब सर के ऊपर से उड़ उड़ जाएगा. So I want all of you all to be very confident with at least this particular, how the 8 nuclei are going to be formed. Okay, now it's very simple. आगे की कहानी बहुत simple है. So just show you here. Yeah. So now this will give rise to what? I'll just show you. Take my video this. Okay, so here you can actually see all of them. Now I'll draw. I have to use a pen. So now you can see I have made 3 cells. So I have drawn the walls over here now. I have completed the wall formation over here, correct? So here is going to be my 1 nucleus, second nucleus, the third nucleus, the fourth nucleus, the fifth one, the sixth one, and the seventh and the eighth. Can you see how the 8 nuclei are now bound by the cell wall? So now they get a wall. Now there's going to be wall formation. Okay. So these three, okay, plus these 3. Huh? Now what is the fun, Now your books are saying that this is anatropous ovule. तो इसीलिए अभी anatropous ovule के हिसाब से है ना, उन्होंने इसको चेंज कर दिया, इसलिए इधर आ जाएगा अपना चलाजल एंड, इधर आ जाएगा अपना माइक्रोपाइलर एंड. ओके अब ये जो 3 है ना यहाँ पर, these 3 will be called as, क्या कहते हैं इसको, एंटीपोडाल सेल। ये जो है यहाँ पर, कैन यू सी दिस, इसमें थोड़ा सा और एक स्ट्रक्चर मुझे ड्रॉ करना पड़ेगा, बिकॉज अदरवाइज इट विल नॉट become, you can see this, I'm drawing like finger like things, है ना, ये finger like things भी मैंने ड्रॉ किए हैं, वो क्या है वो मैं बताऊंगी। So these finger like things ना, 2 are going to be called as your, what do we call them as, synergids. Okay, and here is going to be the egg cell. So what is what I'll tell you, these 1, 2 and 3, all 3 are antipodals. This 1 and this 2 are the synergids. This is going to be the egg cell, only one. And these 2, can you see these? ये 2 न्यूक्लिया बेचारे अकेले हैं इधर, है ना, इनको हम क्या कहते हैं, we call them as, what do we call them as, polar nuclei. So now you understood, ये जो बनाया ना बेटा स्ट्रक्चर, 3 antipodals, 2 polar nuclei, 2 synergids and 1 egg. How many cells you can see over here now? कितने cells आपको दिखाई दे रहे हैं? Can you all tell me? How many cells? 6 cells? All of you can see 6 cells? Or anyone who can see 5 or 7? 7 cells? Okay. Anyone else? 6. Okay. Come on, tell me. Yes, Bhumi, you're right, my dear. You're right. You're right. So I'll write down. Okay. 3 antipodals. Barabar? Correct? 2 synergies. Correct? 1 egg cell. Correct? And 1 which is neglected is the central cell, which is having these 2 polar nuclei. Can you see? Even this is 1 cell only, beta. So totally how many cells? Can you all tell me now? Seven celled, 8 nucleated, and what we call it as now? What we call it, bacha? We call it as embryo sac. So this particular diagram that I have drawn is the diagram of the embryo sac. So what is the embryo sac going to be made up of? 7 celled, 8 nucleated embryo sac. Synergids are present only on one side, yeah, near the micropylar end. 2 synergids near the micropylar end. And 1 egg cell. Okay. अब है ना, ये देखो, ये जो है 2 synergids plus 1 egg cell, इनको हम क्या कहते हैं पता है? We call it as egg apparatus. Yeah, egg cell is always between the 2 synergids, always. Yes. Okay. So this is going to be called as egg apparatus. What is egg apparatus? 2 synergids plus 1 egg cell. So sometimes, you know, to fool you all, what they ask you, that the embryo sac is made up of how many cells? कभी कभी ऐसे पूछते हैं, So you should write 7 cells, right? And kabhi kabhi पूछते हैं the egg apparatus is made up of how many cells? So egg apparatus is only 3 cells, 2 synergids and 1 egg. So don't get confused between the questions, huh? Right? Now understood how the female gamete is going to be formed? Yes. So I'm talking about that only, Bacha. So in the last class, do you remember that I had told you there is a suspense which I'll be clearing it in the next class? वो suspense किसी को याद है? क्या है वो suspense? मैंने बोला था वो serial में कैसे next episode में दिखाते हैं, वैसे मैं आपको next lecture में बताऊंगी कि उसका मतलब क्या है. In the beginning also someone had asked me this question, Miss, are you going to tell this today? And I had promised, yes, I'll tell it today. Recognition, yes, wall held recognition factors, exactly, my dears. So those wall held recognition factors are going to be because of the synergids. The synergids are responsible for this. Okay, so now you understood what is the role of the synergids. So the synergids are the one who are going to involve in this wall, like the wall held recognition factors. So synergids are most important, which are guiding, which are holding the egg also at the center. Now you got the answer. What is the role of the synergids? And also the last class का मेरा जो promise था वो मैंने आपको बताया. Correct? Right, my dear. So I hope till here you all have understood very precisely that how the embryo sac is formed. Okay. But that you will be the same that they had told in the last Sunday ka test. Huh? Okay. Yeah. First 4 chapters also I'm going to this coming week we'll start with those sessions also. Okay. Understood this? Have you all copied down this entire thing, whatever I have drawn on the board? White board के जो भी मैंने डाला है वो आपने कॉपी किया ना बेटा सबने. ये बहुत इंपॉर्टेंट है टू क्लियर द मिसकंसेप्शन नहीं रहेगा बिल्कुल भी फिर. All of you? Very. So I'll just, before winding up for today, just 2 minutes, I'll clear this particular megasporogenesis wala part in 2 minutes. It's 5:32, already late. Why do they have it also? I'll tell you. Okay. So, chalo, fine. We'll talk about this in the next class because it's almost 5:30 na. So next class we can go about with the development of the female gametophyte, but I'll not draw the diagram again because diagrams I have already drawn it today and you all have made a note of it also, right? So development of the female gametophyte onwards, I'll go in the next class. Okay, chalo, bye beta. See you all. Yes, on Friday.